All right, welcome everyone. So today we're going to be talking about multi-phenotype associations. And there's a cool term that's been coined surrounding that field for PheWAS, namely phenome-wide association studies. So instead of associating genetics with other phenotypes, how do we associate every phenotype with every phenotype out there? And I've abused the term a little bit here by basically using it as the title of a section of the lecture, when in fact, it's only one part of the multi-phenotype association that we're going to talk about today. So here we are at the bottom of the 6th inning here. And we have one more lecture to go from the class itself, and then a few wonderful lectures from you guys on the final projects next Tuesday. We talked about cancer genomics, we talked about single-cell genomics, and today we're talking about phenome-wide associations. So first, we're going to start with why multi-phenotype associations, what is the motivation? And then we're going to look at different ways of dealing with multiple phenotypes. On one hand, dealing with GWAS and epigenomics across many phenotypes, looking at the epigenomics of PheWAS, and then looking at meta-phenotype inference and imputation from the clinical record. So first of all, why multi-phenotype analysis? So basically, what are they trying to do? On one hand, association testing has been typically between, genetic variants and a single trait. Every single example we looked at so far, it was basically looking at a single variable and how it varies across hundreds of thousands of individuals, but that one single variable. As opposed to an increasing trend to have really, truly much richer phenotypic data. So instead of measuring a single variable across individuals, what you can do is actually measure hundreds of variables, or perhaps the entire clinical record for those individuals. So that's what PheWAS is all about. It's basically the realization that there's a lot more information out there. So therefore, we can actually use it to good purpose. And what can you do with that? You can basically sort of discover new associations with multiple traits. You can recognize similarities and differences between different phenotypes, and also infer networks of phenotypic variables and genetic variables jointly. So you can use it for many different types of applications. You can basically associate either unknown or underappreciated phenotypes with known ones. You can basically say, well, hey, the activity of the frontal cortex when stimulated with a learning task is in fact correlated with schizophrenia. So that particular trait by itself might be underappreciated, but when you realize that it is in fact a very good predictor of schizophrenia, you can sort of use the two together. You can identify disease mechanisms based on the intermediate phenotypes that mediate them. So this can be at the, from the genetics to the epigenomic level, the transcriptional level, the imaging level, as I mentioned earlier for the brain example in schizophrenia, and all kinds of other endophenotypes that sort of can give you a hint as to sort of how is this leading ultimately to the more complex phenotype. And we're going to see some cases where by relating different types of phenotypes to each other, we can in fact infer causality between them. You can predict disease with easy to measure biomarker phenotypes. So for example, the level of lipids in your blood might be highly indicative of, type 1 diabetes or your obesity predisposition and so on and so forth. And these biomarkers can sort of have a lot more information than genetic data, which usually has very weak effects. Whereas these biomarkers may actually have much stronger correlations and may be much more predictive. Even if they're not causal, they can be helpful biomarkers. You can also use combinations of these phenotypes to impute missing data for many, many more individuals. So basically, in your electronic health record, you may have the variable of interest for only a small number of people, but you may have highly correlated variables. And therefore, you can use these highly correlated variables to impute the missing data across different phenotypic variables, and then improve your GWAS power by imputing the missing phenotypes across the entire cohort. So even if you've only measured that particular lipid level in the blood, to I don't know, 1,000 individuals, you may be able to impute it across 100,000 individuals and therefore boost your power greatly. You might be able to improve the biological relevance of genome-wide association studies by actually carrying an association, not just with the phenotype individually, with each phenotype individually, but with meta-phenotypes that might be more indicative of the true underlying biology, because they combine multiple phenotypic variables in a single umbrella meta-phenotype. Everybody with me so far? Awesome. You could also use it to understand pleiotropic effects. So pleiotropic is a Greek word, and pleio basically means many, kind of like the Pleiades or whatever they call them, Pleiades, which basically have many, many stars together. So pleio means many, and then tropic tropos is the many ways. So pleiotropic effects are basically effects where a particular genetic variant can act in multiple ways, or a particular gene can act in multiple ways. So this is a term that's usually used for the same genetic locus, or the same genetic variant sometimes, or sometimes the same gene through different genetic effects, may actually affect multiple phenotypes. So for example, the same gene might be involved in controlling your metabolism inside your adipose cells, and also your food selection in your brain. And they're seemingly unrelated pathways, but they can converge later on, or simply have the same locus having many different effects in otherwise unrelated and non-convergent phenotypes. You could also guide both experiments as well as diagnoses by predicting missing phenotypes. You could basically say, well, hey, I can predict your lipid levels will be unusual. So why don't you go ahead and take that test, and so on and so forth. So you can basically say, hey, this patient seems to have type 2 diabetes. Let's measure their glucose level in the blood and then see if we can confirm that prediction, which can be made based on many other phenotypes. And you can also start moving towards personalized medicine by basically focusing on combinations of symptoms rather than individual phenotypic variables. Everybody with me so far? So that's sort of, strong motivation for studying multiple phenotypes together. And if you look at electronic health records, they try to achieve exactly that. So basically, electronic health records contain very rich information. They contain, for example, the drugs that a person is taking, the lab tests, such as blood lipids, for example, that the person might have taken. It can contain imaging information from, a brain scan or a, X-ray. And they also contain free text information from the doctor taking notes. And all of that has been put into various databases over the last few years, increasing digitization of these data sets, which can be used in point of care, which can be used to sort of infer statistics across different types of variables, and which can also be used to carry out research. So this information includes clinical notes. So basically these are unstructured free form. These contains lab tests. Basically these are, observations and identifier names and, codes. Billing codes, these ICD-9 codes, International Classification of Disease, ICD-9, ICD-10, are names that you hear very often, which are sort of standardizing the nomenclature so that these can be interoperable across different systems and different hospitals. And another classification is the DRG classification, which is the diagnosis-related groups. Also, there's current procedural terminology, or CPT. So all of these are different billing codes. They were initially used to sort of help insurance companies pay for different things, but more and more they're recognized to actually be very phenotypically informative for the patient themselves. But of course, they have a lot of limitations, which can be partly overcome by treating that data together. And then these can also include pharmaceutical prescriptions. For example, RxNorm can be one such type of database for prescription drugs. Everybody with me so far? So if you look in the last few years, about a decade ago, there was only 10% adoption for electronic health records, and we're now at 90%. I mean, this is a dramatical transformation of the healthcare system in just a very, very short time. And this is sort of the places where there were just simply no, no clinical records, to having it, nearly 100% frequency in all of these places, which is, again, a remarkable transformation. Moreover, in addition to being more prevalent, they're increasingly comprehensive. So, HR data can include many different types of variables, and more they include richer information. So initially, they would include comprehensive data for, much smaller proportion of individuals that even had EHRs. And now it's, roughly 40% of the ones that have EHRs have comprehensive IHRs. And that's something that has continued both in more hospitals having rich information, as well as that information becoming richer. So the electronic health record data itself can be thought of in a Bayesian framework as to being a reflection of the true state of the patient. We never observe the true state. We only measure various things which can tell us about the true underlying state. So basically, the true patient state is something that's hidden from us, but through some recording process, we can have some raw electronic health record data, and that's a reflection of that. But what's really interesting is that the true state also dictates the type of healthcare that the person will go under. So basically, whether a person will be treated or not, we'd like to think, is actually dependent on the true state of that patient, if the doctors are able to do an accurate diagnosis. And that can in turn inform the types of phenotypes that are gathered for those individuals. So if we believe somebody has type 2 diabetes based on other indications, we will go ahead and measure their glucose levels in the blood, for example, as a way to validate our prediction, or to test whether they have it or not. But that might not be something that we test routinely for individuals who are not at risk. And that's sort of a place where that true state can influence both the values of a measurement, as well as whether a measurement is made at all. Everybody with me here? So that basically means that the missing data is actually not missing at random. It is missing in a very particular process, which is actually not at random. And the missing data is more likely to be normal than the observed data, which is more likely to be abnormal. So if I only measure glucose levels for individuals that I expect to be at risk, the distribution that I'm going to get for the observed data is going to be very skewed towards abnormal levels. Everybody with me on that? Great. So then basically, using all that, I can then, develop all kinds of knowledge tasks, like classification of patients into groups, prediction of mortality, or prediction of whether somebody will get sick, and, understanding of the underlying biology, and also guiding interventions for those individuals. Everybody with me so far? So basically, this is a lot of motivations for why we want to look at multiple phenotypic variables at once. So now, let's look at sort of different ways of doing so. Basically, the first is let's use genetic information to model these pleiotropic effects. Here's something that we've touched upon before in terms of the hugely complex architecture of some of these psychiatric disorders. So across 3,000 cases and controls, there's the ability to actually, predict a score associated with psychiatric disorders. And then this score can be associated with additional disorders beyond schizophrenia. So for example, if you predict that score for one trait, you can then sort of turn around and say, well, is this predictive of another trait? And what you find is that, for example, genetic variants associated with schizophrenia are also associated with bipolar disorder, and much more rarely associated with non-psychiatric disorders, suggesting some sort of common architecture and common genetic basis for both. In fact, that's what led to a push for increasing sample sizes when nothing was coming out of these genome-wide association studies, by basically saying, listen, we know there's genetic signal there, let's just push harder and we will try to find it. So doing this basically resulted in many, many different places in the genome associated with disease. But some places are then starting to be multiply associated with disease. So basically, if you look at mapping of genome-wide association study hits across the genome, you basically start seeing some loci that are associated with many, many traits. And that was the first motivation for PheWAS rather than just GWAS. So what is PheWAS doing? Basically, GWAS is asking for every genetic variant, what is its association with a particular trait? What PheWAS is doing is asking, instead of putting different genetic variants on the x-axis and a particular phenotype for each plot, what we're going to do instead is ask for a particular genetic variant, what are all of the traits that it is associated with. So it's actually flipping GWAS around. And it's basically saying, what is the phenome-wide association between a bunch of different phenotypes and a SNP. So instead of asking for the association between a bunch of SNPs and a phenotype, it is asking what is the association between a bunch of phenotypes and a SNP. Okay? Raise your hands if you're with me so far. Awesome, great. So that's what PheWAS is doing. It's basically flipping things around. And you can look for, this particular SNP, which is sitting in this locus, what is its associations, and you can see that suddenly there's a lot of different traits that are lighting up. There's basically both neoplastic traits, such as carcinoma, skin neoplasm, non-melanoma skin cancer, and so on and so forth, as well as dermatological traits, such as keratosis or dermatosis or solar dermatosis, and so on and so forth. So that is basically saying, hey, wait a minute, through this common genetic variant, I can associate skin disorders with melanoma disorders. And of course, biologically, this makes a lot of sense, but it's nice that it's sort of coming out of the data directly. And this can provide a systematic approach to basically look for trait-trait relationships through the genetic variants that they share. Here's another example of leukoplakia and then keratosis. Here's another example where you can see sort of many more cardiovascular traits as well as hematopoietic traits, which are, metabolic traits, which are very closely related to each other. So myocardial infarction, coronary atherosclerosis, as well as type 2 diabetes, for example, are both associated with the same locus. So suddenly you're linking together phenotypic traits that would otherwise be separate and studied in isolation. And here's another example from the HLA locus. This is a lot of where the major histocompatibility complex sits and where a lot of these sort of variants affect the immune system functioning of different individuals. And you can see here that it has a very large number of associations with type 1 diabetes, with rheumatoid arthritis, with all kinds of other traits. Okay. Any questions so far? So you could also do the same across these billing codes, across these electronic health record billing codes. So you can basically look at all of these codes and then ask, how are different traits associated with each other? And you can see here, for example, multiple sclerosis is associated with all kinds of, or co-associated with all kinds of additional traits. So basically the way to think about PheWAS is, to kill many birds with one stone. So basically you have, the single stone, which is a SNP, and then you can look at all of the different birds, the, phenotypes that are associated with them. And you can use that from the very discovery phase. You can basically say, well, if I have multiple phenotypes to start with, instead of thinking of a single phenotypic variable, a single vector of the phenotype for a particular person, let me think of a matrix instead of n individuals, and instead of having only one trait, having d traits at the same time. And then, you can correlate that with the genetic information for those individuals and genotype matrix X, and then the effect sizes of each of these variants on each of these traits. So basically, this becomes a matrix operation for transforming a matrix to a matrix rather than sort of individual vectors for each of those. And then, of course, at the same time, you have this random effect matrix for n by n individuals, and then this environmental component, which can be sort of capturing the genetic variation and the environmental variation. So you can basically model the multiple traits using these multivariate linear mixed models instead of univariate linear mixed models. And when Matthew Stevens and his group did that in Nature Methods in 2014, they basically saw that depending on the number of phenotypes that are affected and the number of traits that are associated with them, you can basically get a huge boost in power by basically doing multi-trait modeling compared to, a single trait model. Basically, if you model 4 traits at a time versus only 2 traits at a time, you can see this, dramatic increase in power and increase of variance explained for every SNP by basically using this multi-phenotype modeling approach. Everybody with me so far? So there are many different ways of doing this. You can basically study the relationship between different traits by basically asking, hey, are they co-associated with the same SNP? Are they, can I sort of boost their power of individual GWAS discovering by using them jointly? Or you can use this LD score regression that we talked about in our heritability lectures to basically calculate the shared genetic component of multiple traits. So in this particular case, you can basically study pairs of traits and then ask, what is the number of individuals, for example, and then what is the LD score for those individuals, which is basically summing up the associations and the correlation patterns for every SNP in that block. And then using that, you can infer the LD score for each SNP across pairs of traits. And then you can use this LD score correlation in estimating the relationship between different traits. So for example, if you look at, if somebody has ever smoked, you can ask what is the co-association at the genetic level using LD score regression with BMI or with childhood obesity or with fasting glucose level or type 2 diabetes. And you can see here how each of these traits is basically showing these significant co-associations. In blue here, you can see groups of traits that are very highly correlated. For example, height, and then the head circumference of an infant, or the length at birth, or the weight at birth, are in fact all very, very highly correlated with each other. And this is basically by asking what is the shared genetic component of these traits across the genome, even if any one of them might not be genome-wide significant, you can still study the correlation and find Crohn's disease and ulcerative colitis, for example, which are both immune traits of the intestine and the gut. HDL, cholesterol, and the age of menarche when the periods start. You can look at BMI and childhood obesity, or fasting glucose and type 2 diabetes, and, depression, bipolar, and schizophrenia. You can see this huge shared genetic component. Everybody with me so far? So you can also do this at the region level. You can basically go region to region and then compute what is the base factor, how much more likely am I to be associated with a disease compared to, by chance, for different models, and then calculate the similarity between traits based on, distinguishing models of sharing or distinct effects. And again, this leads to very similar results of sort of groups of traits. In this particular case, a lot of different hermatological traits and anthropological traits related, anthropometric traits related to each other. And what Pickrell realized, Joe Pickrell, is that there's a very cool thing that you can do with that. You can basically say, if I take all of the variants that are truly associated with BMI, do I then see whether they have an effect on triglycerides? And I can do the same by basically asking, if I take all triglyceride variants, do they have an effect on BMI? And what he found is a very interesting asymmetry between pairs of traits. That basically, in one direction, conditioning, basically ascertaining specifically for BMI, led to a correlation with the effect size on triglycerides, Whereas in the opposite direction, if I only look at triglyceride-associated variants, there was no correlation between their effect size on triglycerides and their effect size on BMI. And that basically led him to postulate that, in fact, that was evidence of directionality, of causality. That basically, if BMI variants truly have an effect on triglycerides, then there will be a strong correlation of the effect sizes when I focus on BMI variants, but no correlation when I focus on triglycerides. And similarly for LDL cholesterol and coronary artery disease. You can basically see that focusing on one leads to correlation, focusing on the other loses that correlation. So that allows us to now start doing causality, causal inference from the unidirectionality in these conditional associations. Who's with me so far? Here are some additional examples. So BMI has an effect on type 2 diabetes, but not the other way around. So basically, again, BMI is a huge risk factor, body mass index, huge risk factor for type 2 diabetes, but type 2 diabetes does not lead to obesity. And conversely, having greater than normal thyroid activity basically is associated with height, but not the other way, with inversely with height, but not the other way around. So you can actually use that to start inferring pleiotropic variants by basically looking at every combination of settings between pairs of SNPs and their alleles, and then asking what is the corresponding effect size on each of the different phenotypes by enumerating all the combinations. The other approach is to basically start asking about partitioning of the genome into its phenotypic associations. So the concept here is that we can now start doing genetics at the systems level, basically looking at the whole genome each time and many, many different phenotypic associations. So what systems genetics tries to do is basically characterize multiple polygenic associations for complex disorders, basically define what is the normal range, define the correlation between genetic variation and environmental stress. And this was quite a pioneering paper by Joe Nadeau, proposed more than 7 years ago, basically start understanding the comorbidity patterns, co-association patterns of different traits. More recently, there's several people who are still wondering, hey, what has come of that field? Basically, where is system genetics today? And there's still a lot of debate about that. So basically, sure, we can characterize systems level genetic outcomes, just not one gene at a time. And then we can integrate data across many different modalities. We're still missing a lot of sort of the inference of networks and genes and correlations. But now with larger and larger biobanks that have genotyped individuals across thousands of traits, each profile for thousands of traits, we can actually start going a step beyond that and truly deliver on the promise of system genetics. So in the UK Biobank, for example, we can look at hundreds of different traits, and in this particular case, we're selecting 47 high robustness ones that Alkis Price and Ben Neal and others have been helping to process and disseminate. And you can see here some of these different categories of traits. For example, height associated, hair color, platelet counts, bone mineral density, blood pressure, education, and so on and so forth. And what you can actually do is carry out a phenome-wide association study by effectively clustering the genome. You can take every genetic region and then ask, what are the traits that this region is associated with? And you can then look for the vector of trait associations for every region of the genome and cluster those regions that have similar associations. So you can estimate the genetic heritability based on summary-based QTL modeling for every locus, and then ask if they're strongly genetic with respect to the reference genome panel, then identify causal SNPs from multivariate effects across thousands of LD blocks in the European population. And then what you can construct is a matrix that tells you for every LD block across these 1,700 blocks, what is the vector of association across the 47 traits. and what are the causal variants within these blocks. And then you can cluster the LD blocks according to their trait association. Who's with me so far? Awesome. So that's what you can do. You can basically first estimate the multivariate genetic effects from the univariate GWAS statistic. So, you can have the phenotype of each individual, and then the genotype of each individual, and then the univariate GWAS effect, and then some kind of standard error, and then the genetic effect for all SNPs. And then you can estimate this theta, which basically tells you what is the effect on all of the phenotypic traits jointly. So you can basically find map causal variance, taking care of the LD, and then doing the univariate stats, correcting for that LD inflation, and then estimate a heritability for every genetic variant in the set of traits. And you can force this to be sparse. So basically, you can basically have a summary-based model that doesn't require to actually have the full genotype matrix for all these individuals, by basically looking at the summary statistics rather than the individual per individual association. So when you do that, you basically find that different traits are basically very highly heritable using this linear model of common genetic variation. You can see here that the mean platelet volume, for example, is 60% heritable. Bone density is 40% heritable, and the height is 54% heritable. And that actually captures a lot of that heritability. So basically, as you start going through this exercise, you basically see that you can start capturing a much larger fraction of the heritability than what we had previously ascertained, because you can now go to much more unbiased cohorts, larger sample sizes, and sort of this multi-phenotype approach, which can be much more robust. So what you can basically ask is, what are the causal variants in each of these loci that captures almost all the genetic variants? So basically, you don't take one genetic variant per locus, you basically take more and more and more until you capture, a big chunk of the genetic variants of that locus. And that goes directly at that pleiotropy and polygenicity aspect that I talked about. Again, no single SNP dominates the amount of genetic variants explained. And this includes both additive and constitutive effects. And it turns out that these sparse effects of these common SNPs can capture almost all of the heritability. So instead of worrying about rare variants that might be hidden within these haplotype blocks, we in fact use multiple common variants within these blocks, and we find that they do capture most of the heritability. So the question is, does this very high heritability necessarily imply this omnigenic model where everything has an effect on everything? So we talked about omnigenicity before, namely that all SNPs contribute to the heritability of every trait. And, that has posed a lot of sort of philosophical challenges to the GWAS mentality itself. When we were initially saying, oh, GWAS is great because it's going to tell you about what are the pathways that underlie disease. And Joe Pickrell basically says, well, hey, wait a minute, if everything's associated with everything to varying degrees, previously we're pretty excited to have a genetic effect from this pathway to that phenotype. Right now, if every phenotype is associated with every pathway, that makes it much harder to justify this notion of prioritizing pathways. And but there's another way to look at that, which is, yes, certainly, as I keep going down the list of loci, I can explain more and more and more and more heritability. But then the fraction of the heritability that remains to be explained is actually fairly small. And if I start including more and more of these LD blocks, I capture most of the heritability with relatively few blocks. And then, of course, there's a very, very long tail, which I think was what prompted the omnigenic model. But the high concentration of heritability on a small number of LD blocks is, in fact, instead arguing for this very strong concentration of heritability into a small number of pathways. Everybody with me so far? So you can do this basically across many different phenotypes. And when you do that in the UK Biobank or when you do that in the Japan Biobank, what you find is that for every one of 47 traits here and 58 traits there, you basically find this extreme concentration of the heritability with this very strong inflection point, after which there's very small diminishing returns, but before which every single LD block is capturing a large number, a large fraction of the heritability. Any questions so far? So you can then ask, great, I've now clustered my genome according to the co-association patterns across the many phenotypes of every single LD block. Let's now try to color these blocks based on what are the corresponding epigenomic enrichments for each of these blocks. So basically what I'm going to do is partition the genome into LD blocks. For every LD block, look at its association with many, many different traits, cluster blocks that have similar associations, and then say, hey, do they show an enrichment in similar epigenomic annotations? And the answer is overwhelmingly yes. Basically, if you look at these clusters of LD blocks, they're say 25 clusters here, and different blocks are associated with different combinations of phenotypes. For example, block number 3 here is associated with all of these blood phenotypes. So white blood cell count, platelet count, lymphocyte count, red blood cell count, and so on and so forth. And then all of them appear to be enriched in blood enhancers, which is kind of cool. But basically, it sort of says that, hey, if I cluster these blocks according to their phenotypic association, they basically, are biologically consistent. If you look at the years of education or college or any of that, you basically find that they are associated with these clusters here, and then they are ultimately having an effect on the central nervous system, CNV, over here in green, and so on and so forth. Basically, what that suggests is that we can, in fact, infer a lot of distinct biology for the genome using not just one trait at a time, but having this really, truly systems genetics approach, where for every single locus, you're asking what is the complete set of association, and then clustering the loci, not just based on one trait at a time, but based on all of the traits they're associated with. Everybody with me so far? Awesome. So you can do this by basically first saying, let's cluster everything based on these associations, but that requires that, number one, you have associations to start with, and basically the signal at any one locus is sufficiently strong. And number two, it only clusters things after the initial association. An alternative to do that is to basically use factorization from the beginning. So instead of basically first partitioning the loci and then clustering them based on their associations, what you can basically do is actually start factorizing the association matrix into components by basically, breaking up that matrix using linear algebra into multiple factors. So basically, instead of just saying what are the associations with each trait individually, we're going to effectively define groups of traits that are co-associated as factors in a factorization framework, as factors of this association. So basically what you're actually doing is jointly partitioning both the genetic variance, the genetic loci, and the phenotypic associations of those loci by basically taking this very, very large matrix of genotype by phenotype and then partitioning it into different factors. So basically your effects, lambda, are factor specific. And then, both the trait effects and the genetic effects are sort of partitioned in that way. You basically take your univariate GWAS effect matrix from the biobank. You basically build this reference LD matrix R from a matching cohort. And then you study the effect of every SNP on each factor. And then the factor could be, for example, psychiatric traits, and that could include schizophrenia and bipolar, or immune traits, and that could include rheumatoid arthritis and multiple sclerosis, and so on and so forth. Everybody with me so far? So using this, instead of now clustering loci, we are clustering factors. These are sort of these cross-cutting effects. And you can basically sort of start grouping together these UK Biobank traits across the factors that are associated with each trait. Basically, this is the same idea of partitioning your genetic associations, but instead of doing it discretely based on loci, you're now doing it, in this cross-cutting fashion across factors. And you can then take that matrix and then say, well, for every trait, what is the co-association of that trait with every other trait through the common factors? You can basically turn this factor by trait matrix into a trait by trait similarity matrix. And that allows you to, again, capture the same groups of phenotypic variables as we saw before. Who feels that they've learned something? Awesome. Good. Any questions? So basically doing this, you can basically reveal localized patterns of pleiotropy and an explicit selection of causal SNPs within each locus. Here's one example where, as we're going through the locus, we can basically find the association with different traits. And then, conventional PheWAS method would only highlight a single SNP in this particular approach. And the SNP by SNP methods would basically be inflated by LD, and they would not be so exact. The PheWAS tests would basically have a huge number of multiple hypotheses burden, because you're testing every SNP against every trait. Whereas factor GWAS basically dramatically reduces the number of parameters using this factorization approach. And this is all work that's led by Youngjin Park as a postdoc in the group. So basically, this is what he ends up with. He basically can factorize GWAS and then resolve 94 clusters of genomic regions based on their pleiotropic patterns. And then these are the genomic loci where these factors can now project. And these are the co-associations of these. For example, you can see respiratory disease and asthma are sort of co-clustered. You can see allergy and eczema for some of these factors, but not all. You can see here, pigmentation in hair and pigmentation in the body, tanning, sunburn, and all of that. You can basically see how these are starting to be partitioned. And for each of those, we now have the ability to study them biologically. So then for each of these factors, you can basically ask, what is the total number of causal SNPs that I'm predicting to be associated here? And you can see that, for some of these factors, it is hundreds of SNPs, and for others, it is fewer. You can say, what are the total number of SNPs, including non-causal SNPs, everything that's in LD with these factors? And then what is the total number of LD blocks? Basically, these are independent genetic contributors to each of these factors. So you can see here that you're clustering hundreds of LD blocks. In some cases, all with the same groups of multiple. And then this is just a zoom in of this particular matrix. So basically, we can use this co-association to basically identify traits that are frequently sharing common genetic factors. And this is what this looks like. So you can basically start clustering, hair and skin, balding, different metrics of that. Asthma and respiratory disorders, lymphocyte versus, white blood cell counts and platelet counts and hemoglobin and cardiovascular traits and so on and so forth. Everybody with me so far? And then here some of the clusters basically zooming in. One cluster has lung capacity, cardiovascular disease, respiratory disorders. Here's another cluster that has, pigment hair, BMI, and so forth. So basically, that's sort of another way to carry this out. Everybody with me? So what we're going to do now is change gears a little bit and basically start looking at the epigenomic enrichments of these multiple trait associations. So we mentioned earlier that we can basically measure the epigenomic landscape across many different tissues and cell types, as we saw in the Epigenomics lecture. So the Roadmap Epigenomics Consortium has basically generated these annotations across the whole genome for epigenomic variables. You can then now start asking for each of the associations in any particular trait, what are the epigenomic annotations underlying this? And you can do these kinds of enrichments that we talked about earlier, of basically finding what are the genetic associations that are systematically localizing in similar epigenomic traits across the whole epigenomic annotations across the whole genome. And we previously did that one trait at a time. We basically said, hey, what is the association of all schizophrenia-associated SNPs with different H3K27 acetylation peaks? And we found that, for example, the central nervous system was capturing a lot of that. But you can also do that by combining multiple traits simultaneously. So here's one example where in the same locus, you basically have multiple traits being studied jointly, basically prioritize across the genome these annotations that are the most strongly associated with these multiple traits, and the SNPs that are then prioritized within these enriched annotations. And you can see here, for example, if you combine multiple psychiatric traits like neuroticism, years of education, schizophrenia, basically have this very strong enrichment for CNS. And if you start combining Crohn's disease, multiple sclerosis, and type 1 diabetes, again, you have this strong enrichment for immune traits. So what you can basically do is, in the same framework that we talked about before, of having an epigenomic annotation that gives you a genome-wide enrichment for SNPs in that trait, which then gives an empirical prior, what you can basically do is add a disease covariance factor that is basically telling you about the co-enrichment of multiple traits on the same set of epigenomic annotations. So instead of treating every trait in isolation, you now can combine the multiple traits to basically ask about their patterns of co-enrichment, which can then lead to a much more robust measure of the enrichment of any one trait. So basically, when you do that, you basically find that this allows you to combine the traits and cluster them according to whether they have neuropsychiatric effects or neurodegenerative effects or immune effects or cardiac effects and so on and so forth. And then for each of those, cluster the corresponding associations according to whether they show epigenomic enrichments for immune cells, gastrointestinal cells, central nervous systems, and so forth. And you do generally find a very strong agreement between the two, where traits that have similar biological properties are enriched in cell types that have matching biological properties. Everybody with me on this? So then you can do that to basically guide the cell type specific enrichments by first studying these enrichments at the group level and then projecting them down to the individual cell type level. And what you can basically see is that very similar traits are in fact correlated with very similar epigenomic enrichments. So you can see here ADHD and neuroticism, schizophrenia, years of education, and so on and so forth are basically showing similar co-enrichments, which makes a lot of sense in terms of their association with the psychiatric and central nervous system epigenomic annotations. And then here you can see LDL, HDL, triglycerides. They're all associated with metabolic tissues. And then this group here of celiac disease, Crohn's disease, lupus, ulcerative colitis are all associated with each other and also all co-enriched in immune processes. So if you basically do your association and epigenomic enrichments one trait at a time, you basically can get pretty strong EQTL enrichment. But if you do it multiple traits at a time, the enrichment for EQTLs is in fact increasing in every single case, suggesting that the joint application of multiple traits simultaneously is increasing your robustness and making the signal better. The Psychiatric Genomics Consortium basically took a similar system genetics approach to basically study co-enrichment at the pathway level. So basically gathering pathways from public databases, pruning the redundancy, and then asking what is the association for genetic variants in that pathway rather than genetic variants in isolation. And then combining these enrichments basically improved the causal pathway detection. So, you know, any one trait in isolation was insufficiently enriched, but when you combine multiple traits by sort of working with the Psychiatric Genomics Consortium, you basically see a lot of these pathways are now becoming genome-wide. So we basically looked at many different ways of combining multiple phenotypes together. We basically saw this concept of clustering the genome into their co-association patterns, into directly factorizing these enrichments for different associations, and also partitioning the genetic signal into the specific functional categories across multiple traits at a time. The last part that I want to talk about today is the concept of inferring these meta-phenotypes and then imputing individual phenotypic variables using those meta-phenotypes. So in this particular case, we're not going to focus on genetics. We're only going to focus on the phenotypes that come straight from the electronic health record. And the reason for that is there are truly millions of individuals in different hospital systems, such as, you know, the Mayo Clinic, the Partners Healthcare here, the Geisinger system, Kaiser Permanente. All of these are, in fact, containing millions of individuals, but only a small fraction of those have been genotyped. So basically, we're looking at, 10,000, 50,000, 100,000 for some of the largest cohorts, but still nowhere near the millions. So the genotypes are often not available over the large patient cohorts, but given the causal mediating phenotypes, diseases of interest are conditionally independent of the genotype. So electronic health records basically allow deep phenotyping to basically start inferring what are the true underlying biological roots. So the idea here is that if you had such an intelligent system, you could basically sort of, train the model based on existing data and then use it to make clinical recommendations for different systems. You could recommend lab tests, you could recommend specific diagnosis to the doctor that could then be verified by the professionals, or you could recommend prescriptions and even treatment procedures if you could do that systematically. So this is what we've been trying to do. So in this particular case, you can see the comorbidity patterns of different phenotypes. This is a plot generated by Jose Davila in collaboration with ULE. And for each of those modules of comorbidity patterns, you can basically see what are the biological terms associated with the corresponding ICD-9 codes. And you can see here sort of, in a color-coded fashion, how these are clustering with each other in terms of their comorbidity patterns. So then the intuition here is that we're going to be factorizing that matrix. So basically across individuals and across phenotypes, we're going to be basically looking for a lower dimensional representation of these associations, which can then allow us to infer these latent factors. And these can correspond to both patient-to-patient similarities, so clusters of patients that have similar biological phenotypic variables, as well as clustering of phenotypes together when they have association with similar sets of patients. So then the idea here is that we can sort of move in any one of these dimensions of these multidimensional objects. On one hand, you can basically look at the similarity of different EHR features and then the underlying latent disease topics. So these are the meta-phenotypes, which we can learn from multiple combinations of electronic health records, and also the patient dimensionality here. So basically, by looking at the correlation between different EHR features across patients, we can infer latent disease topics. And similarly, by looking at the correlation between patients across EHR features, we can infer patient groups that are more robust than the information for any one patient. So a very common approach in this particular methodology is to actually group words by their topic, so bags of words kind of approaches. Basically say, all these different words are associated with the arts. All these different words are associated with the budget. All these different words are associated with children or with education. And by basically simply counting how frequently are each of these words appearing in a particular document, you can then classify documents according to their topic. Everybody with me here? So basically what we're going to do is a very similar approach where we're now going to learn healthcare topics associated with different words and then infer the disease topic of each person based on the set of words that their clinical record contains. There's many challenges. The first challenge is that electronic health record data are extremely noisy and they're also extremely biased. So unlike corpus documents in text mining, clinical notes are full of typos, are full of arbitrary abbreviations. So here, you can see hypertension and then coronary atherosclerosis, native, CHF, NB, observe, suspect, infer. I mean, these are just, extremely, basically arbitrary abbreviations of, the different notes. If you look at the clinical notes themselves, you can see continue, hospit, medio, and so on and so forth. So basically text mining is usually used to remove stop words. Here, there's no clearly defined stop words. And then the billing codes are not meant to be disease specific. They're just used for billing. So how do you deal with all that? So one way to do it, and the second challenge is that the EHR data are extremely sparse. If you basically ask what fraction of patients has which phenotypes, you see that the vast majority of phenotypes are found in less than, each in less than 5% of the patients. You have basically 30,000 phenotypes that are each associated with, less than 5% of the patients. And then you have some phenotypes that are associated with a huge, huge number of patients. And same for ICD-9 codes, labs, and so on and so forth. So that basically means that somehow we have to deal with this extreme sparsity, where most of the data is simply never ascertained. Basically, most of these variables are simply empty in many of the individuals. Everybody with me for challenge number 2? And then challenge number 3 is that missing data is extremely biased. So basically, if you look at the fraction of individuals for which the particular variable was measured, for example, whether the lab result was taken, you see that so these are different lab tests, 192 lab tests. Each column is one lab test. And then you're asking what fraction of people showed a normal score versus what fraction of people were in the top 1% or the bottom 1%. And you can see here that 20% of the people are in the top 1% and another 20% of the people are in the bottom 1%. How is that possible? That's possible because we are pretty good at predicting whether something is normal or not. And therefore, when a test is prescribed, most of the time we expect to see an abnormal result. And therefore, when I see that these 20% of the patients had in fact an abnormally high and an abnormally low test, that basically means that I cannot use the observed data as a way to build the true underlying distribution of different variables, because then I would be greatly amplifying the extremes of the distribution. Everybody with me here? So, what do we need to do? Basically, what we need is, just different ways of specifically dealing with the non-missing at random problem. So non-missing at random is something that has been, well recognized. If you look at, for example, the users that select music ratings, what they basically, if you look at the users, you basically see this. If you ask randomly, what should my score be? What you will find is that very few people give the best score. In fact, if you randomly assign music to people, you will see that most of the time they're not going to like what they listen to. And then some of the time they're going to like it a lot. But if you look now at the music that people actually listen to, they self-select songs that they expect to like. So then they end up with a much higher proportion of 5 stars and 4 stars and 3 stars than what you would expect if you were assigning these randomly. Everybody with me so far? So that basically means that if the data is missing, that usually means that they're not going to like that song because they haven't even bothered playing that song. Does that make sense? So in the same way with clinical data, if something is missing, very likely to be normal, whereas if something is observed, it's much more likely to be abnormal. And in this particular case, the observed data is very frequently abnormal. It's sort of, really, really like the song, I'm more likely to observe it to start with. So there's been a lot of work in sort of explicitly modeling the pattern of missingness, of basically studying the driver mechanism underlying this missingness pattern. So the concept here is that you can actually distinguish different missing mechanisms. So you can basically have a mechanism of missing completely at random, whereby the data is not, the missing data is neither driven by the observed data nor driven by the missing data. They're simply missing at random, which basically says that the missing indicator only depends on the observed results, but not on the missing results themselves. And then there's also non-missing at random, where we have to actually explicitly model both the missing and the non-missing data. So that basically means that the value of the missing data is in fact influencing whether the data is missing or not. So as we saw before, the tests that are missing are more likely to be normal, and the tests that are observed are more likely to be abnormal. Everybody with me so far? So basically, if you look at these model parameters, basically this is, you can explicitly create a variable associating with missingness. You can basically say, here's an indicator variable that will tell me whether the data is missing or not. And then I can have a partially observed result. Knowing what cluster the patient belongs in can then teach me both about the true underlying value and the probability that I will even observe that variable. And that's the whole concept of basically first grouping the patients into clusters. We can then learn which clusters are associated with abnormal scores, and then do a cluster conditional estimation of the values for specific lab tests, so that if the lab test is observed, that will allow me to sort of cluster the patients based on all of the phenotypic information for those patients, but also the lab test itself. So I can start clustering the patients. And then within that cluster, I can sort of say, well, the probability with which the variable is missing is in fact highly dependent on the cluster membership. Raise your hands if you're with me. Awesome, great. So then the key idea is to sort of code the missing indicator as part of your model. You could have the missing indicator as part of your data itself, or you could simply have it as a latent variable within the model. And here you can see that, when you look at overall prediction of medical prescriptions, for example, you can see how many variables do I include missing this information for. You can see here that, up to 5 variables or so. there's continued improvement in the overall prediction based on the area under the receiver operating curve, which basically suggests that indeed modeling the missingness pattern is extremely important. Okay. So then I can jointly model both the lab test assignment, whether it was prescribed a particular lab test, regardless of its value, and what the actual result of that lab test was. So for example, if I belong in a set of patients with respiratory problems, I'm much more likely to be asked to have a complete blood cell count. And that complete blood cell count is much more likely to be abnormal, and same for C-reactive protein. If I have kidney problems, similarly, I'm much more likely to be prescribed a urea nitrogen test than, if I don't. So I can actually start modeling this by sort of having the patient cluster dictate both the lab result and the lab presence itself, and having the lab presence dependent on the lab result. But of course, I don't observe that directly. But knowing the probability of the patient belonging in that particular cluster, I can actually flow information on whether the lab was prescribed or not. So that's basically an example of explicitly modeling the missing mechanism. Basically says, was this lab observed or not? Sounds good? So then, we can do this across many different phenotypic variables. So basically using both lab presence and lab results, I can basically learn hyperparameters that sort of dictate the probability that, particular lab variable was observed or not, and the value of that variable. So then we can jointly model these missing indicators and the lab test results, whereby the observation for a particular patient depends on the score with which that particular patient belongs in the particular topic, the score likelihood for that particular question of that lab test, and the missing indicator likelihood for that particular lab test. So then it basically says that the number of patients assigned to a particular topic when L is observed versus when L is not observed are in fact highly dependent on that class that the patient belongs to. So then you can do that both for lab tests and for all of the other phenotypes. So basically looking at, various binary clinical features, looking at, all kinds of ICD-9 codes and et cetera. You can basically start modeling, number one, the patient meta-phenotype, the class, the topic, or the disease topic that the patient belongs in. Basically, what is their phenotypic class, their meta-phenotype, and conditional on that class, both the frequency of the different phenotypes and whether they are taking a particular test and what the result of that test will be. Sounds good? So basically, I can use this relatively simpler framework for all of the phenotypes, and then this relatively more complex framework for the labs because of the prescription bias for the lab itself. So then what Yue Li did in the group is he developed this model called Mix EHR for this mixture model for electronic health records that now allows you to do exactly what was in the previous slide. Basically, model this meta-phenotype and then treat this missing indicator for each of the variables, as well as all of the additional phenotypic classes. So what he does is that he decomposes the matrix of patients by lab test, by ICD-9 billing code, by questionnaire, prescription, treatment, and all of the clinical notes. And then for each of these different data modalities, he has a particular factorization and then a loading matrix that allows you to combine these different modalities together. So then he can predict the risk for each patient to belong in each class of meta-phenotypes, and then infer the the disease class for each person. So cutting across this, you can basically see DRG codes, ICD-9 codes, lab prescriptions, clinical notes, and medical prescriptions. And then sort of have this hierarchical model that allows you to, at the top level, infer the meta-phenotype. And then within each of the data modalities, infer the sort of topic-specific meta-phenotype. Again, partitioning this phenotypic matrix into each of those components. So what you can do is then start asking in simulation whether the model is in fact capturing, simulated data. And what you can see is that the non-missing at random component is in fact greatly outperforming the missing at random model. So basically, if we assume that the random data is randomly missing, that the missing data is randomly missing, then we do a much worse performance. Whereas if we assume that the data is not missing at random, then we do a much better performance. And then, it agrees with the true model much, much more frequently than you would expect. Here's some terms, for example, that are very heavily associated with different meta-phenotypes. So basically here you're learning the different meta-phenotypes, and then what you can ask is what were the individual disease phenotypes that were associated with that. And what you can see is that for every one of these meta-phenotypes, there's a large number of different data modalities that contributed to this. So that basically suggests that I can combine information from clinical notes, from labs, from prescriptions, from billing codes, from all of these different data modalities is shown here in different colors. You can also ask, how are these topics correlated with quantitative variables such as the age of the individual? And we can basically ask is, what are the topics that are the most positively correlated with age? And what are the topics that are the least positively correlated with, the most negatively correlated with age? And what you can see is that, for example, heart failure, cardiovascular disease, and dementia are three of the most positively correlated with age, which are diseases of old age, obviously. And then if you look at the most negative correlation with age, again, these are, neonate birth and, preterm birth and so forth, which are, with the earliest age group. And, what's really interesting is that preterm is, in fact, even more negatively associated than, say, neonate, which is, again, fully consistent with the biology. You can also ask for different classes of traits. For example, for schizophrenia-associated traits or PTSD-associated traits, what are the top scoring terms? And here you can see, for example, from the clinical notes, delusion and schizophrenia and bipolar and cogentin and psych misspelled and psychot and etc. are basically featuring in the clinical notes of the doctors that are dealing with these patients with patients in this cluster. The ICD-9 billing codes seem to agree with that, and in fact, with schizophrenia, you also see poison tranquilizer, for example, or toxic effect, caustic, no other detail, schizoaffective disorder, and so on and so forth. So you can see here how across different data modalities, we're combining terms that are all strongly indicative of schizophrenia or of post-traumatic stress disorder. Everybody with me so far? So what you can also do is start prioritizing patients based on the disease mixture. So here, for example, you can see that there's only a subset of the individuals who are in this particular cluster that were tagged with leukemia or with pulmonary embolism or with cirrhosis, liver cirrhosis. You can see that there's like gray squares here where that particular phenotype was not explicitly declared. And if you look here, you can see why these patients were in fact selected to be part of that group. And you can see here that even though they did not have leukemia explicitly mentioned, they had all these other phenotypes that are highly indicative of leukemia. Here, even though these guys didn't have pulmonary embolism explicitly mentioned, they have all these other phenotypes that are highly predictive of that. And then you can actually explicitly see what are the terms color-coded according to the evidence type that are associated with each of these clusters. And in cluster M31, for example, you can see, what are the most informative terms in this word cloud. And similarly for topic 35 and for topic 50, you can see that there's many different types of lines of evidence, which are all combined together to infer the membership of these patients, and therefore to complete what would otherwise be a missing informative variable. So that allows us to now start imputing missing EHR codes. So, we can go from something that's extremely sparse to actually imputing the values for each of those. And you can see here that many of the white gaps are getting filled in, suggesting that, in fact, maybe the doctors should have added that additional term. And you can also ask, depending on, basically, you could hide some of these variables. Basically say, well, I know that the patient has that, but I'm going to remove that code. Am I able to recover this? And the answer is, overwhelmingly yes. So basically 82% of the time, if you remove a term, we're able to just, predict it back. And in fact, this is also heavily biased because, usually when you add one particular term, that might be the only one you bother marking. You may not mark it anywhere else. And then removing that one term can in fact lead to a decreased membership in that particular class. The other thing you can do is in fact predict future mortality. So based on the visits today, will the patient die the next time they come to the hospital? That can be actually very helpful. And the answer is that overwhelmingly yes. So basically you have 85% AUC of actually predicting whether a patient will in fact pass the next time they visit the hospital. So, if we can do this reliably, we probably want to let people know and, warn them in advance. Don't wait until your next visit. Maybe you should be, treated. And you can also ask what are the terms that are the most predictive of mortality. You can basically see that, if you have ventricular fibrillation, for example, or, cardiac arrest at the last visits, or anoxic brain damage, and so on and so forth, all of these, you're, in pretty bad shape for making it through your next visit, or renal failure, acute necrosis of liver, and so on and so forth. Or if you're in a mechanical ventilator, it basically is a good indication that the next time around you won't be able to make it, and so on and so forth. So basically what this is, basically telling us is that we can actually make not just classification of the current state of the patient, but also prediction of the future state of the patient. So just to summarize this part, basically these phenome-wide association studies can elucidate phenotypic the networks by linking genetics to their co-associated traits. You can model multiple traits jointly and reveal the genetic correlations between them. And then you can start modeling EHR data, recognize that it's extremely rich, but there's many challenges in modeling it because of the high dimensionality, high sparsity, and the non-missing at random. And then these machine learning methods are especially helpful, and especially generative models. And they hold great promise to learn, compress latent dimensions of these super high dimensional data sets, and also impute the missing data to reveal the underlying network. So this is what we covered today. We basically motivated why we even care about these multiple phenotypes studied jointly. And I introduced the concept of PheWAS, of basically associating every phenotype with every other phenotype rather than every genetic variant with every other phenotype in the context of specific genetic variants. We expanded that to start looking at LD blocks and their co-association patterns across an entire clinical record, such as from the UK Biobank. We went a step further from these region by region co-association to a factorization of the genotype-phenotype correlation matrix to basically understand the different factors of these disease associations. We looked at our epigenomic enrichment analysis that we had seen before, but now in the context of multiple epigenomic annotations and multiple phenotypic variables simultaneously, by basically learning the co-enrichment patterns of specific phenotypic classes into similar epigenomic annotations, and then using that to improve the enrichment and improve the pathway inferences. And then lastly, how we can model these electronic health records by specifically looking at meta-phenotypes, basically these higher order terms that can combine information from multiple individual data modalities, such as clinical notes, lab tests, prescriptions, ICD-9 billing code, and so on and so forth, and how we can in fact use that to impute missing data and to also prioritize patient disease risk. Feels that they've learned stuff? Hey, awesome. One more lecture to go. So on Thursday, we're going to talk about CRISPR and genome editing and synthetic biology and all of that. And then next week, your presentations. Thank you.